Hello, I'm James Graham, CEO of Recce Pharmaceuticals Limited. Today we talk about the new frontiers in sepsis and urosepsis treatment, synthetic antibiotics to combat superbugs. I'd like to thank Sepsis Alliance, of course, for this opportunity and really look forward to getting into this compelling data in the fight against superbugs. Usual disclaimer. Who is Recce? Firstly, where do we come from? So, as you can tell by my accent, we're a little Australian company building a global presence, particularly there in the United States. And we're developing a whole new class of synthetic anti-infectives with unique mechanisms to treat serious and life-threatening infections, particularly sepsis. We have multiple indications in the phase 1, phase 2, and even a registrational phase 3 study coming online, as the World Health Organization has recently added Recce in their list of global antibiotic new compounds. In fact, we are the only in a particular category, and in fact, to my eyes, the most advanced globally across all those noted. We also have a qualified infectious disease designation awarded by the US FDA. That's particular for our sepsis candidate because it's actually defined as bacteremia, which is the broad spectrum nature of sepsis. And clinically, I believe we are the most clinically advanced in that avenue. So brief background of course, me and the team. I'm the CEO and Managing Director. I started as the founding investor, really supporting my grandfather, who was the inventor of this compound, former head of Johnson & Johnson Research Australasia of about 10 years. And we've since joined the team and board, our executive chairman, whose doctorate is in the study of bacteria and blood-borne diseases, Dr. Alan Dunton, who led Johnson & Johnson Research, or Janssen, globally, Michelle Delizia, and Dr. Justin Ward. So urinary tract infections are in fact 25% of the cause of overall septic infections themselves. So where we look to a sepsis patient, 25% have that underlying bacterial infection. 90% of those bacterial infections are generated from E. coli infections. And over 90% of the UTI causing bacteria are resistant to at least one antibiotic. And close to 80% are resistant to at least two antibiotics. So it's one thing to identify it, but you really need a compelling active drug to combat that bacterial infection, whether it be the underlying urinary tract infection through to the all-out septic patient. So what is the challenge we look at? When every other antibiotic is created, the antibiotics start to fail. So here we have on the top line of where the antibiotic was introduced, and in the bottom line where we see resistance starting to take place. And you'll see really over the period of time as we move towards our time horizon of now, resistance occurs quickly. And it really occurs quickly, often because we're using the same theme of the underlying mechanisms of that antibiotic, whilst the bacteria are very familiar with it and already have defenses enabled. This, of course, is looking to the time ahead. So this is the global antibiotic pipeline according to World Health. And some 70-ish% there is doing the same thing, expecting a different answer in the beta-lactams, by example. And the other ones are really also variations of a theme. For ATP production disruptors, which is us, we are clinically the only candidate in this position at this time, and really across all as a new class of antibiotic, I think, as we say here, is recognised as a novel treatment for a range of life-threatening and drug-resistant bacteria, and is the only compound that has a unique mechanism of action able to overcome the energetic component of bacterial species. So why is it so challenging to create new antibiotics, and where have the others really gone wrong? Well, they have specific mechanisms of action. That means, what works today, the bacteria create a defense to overcome it, and it no longer works tomorrow. They're naturally derived, so they're as good as what you find, which of course, without an inventive step in that, it's just a known mechanism that's found under a rock or in the garden, so to speak. And there's other challenge, of course, from corporately, antibiotics are greatly undervalued. So the motivations to develop new ones perhaps might not have been there. improper use and over prescribing. And of course, as a result of this, there hasn't been any new antibiotics approved in over 30 years. I actually think it's over 40 years, but we'll go with the stat here. So here comes a new class of antibiotic. Now, to my knowledge, we are clinically the most advanced new class of antibiotic capable against the full suite of escape pathogens. That's the acronym, of course, of the deadly bacterial species. Why and how is that possible? We have a universal mechanism of action. So what that means is we don't have singular or a couple, we have dozens of mechanisms of action taking place at any one time so that the bacteria can mutate to survive. It always has, it always will, but it will never be able to mutate to a degree that can overcome the mechanisms of action of attack. We work extremely quickly. So we're actually measured in minutes, not hours, of having an antibacterial effect. And we have a broad spectrum capability. So we're not just gram positive, we're both gram positive, gram negative, and their hypercellularly mutated forms. Our compound is soluble at the pHs of the blood to the opposite acidity of the stomach, which means multiple formulations depending on where the underlying infection is, whether it be an IV formulation, topical, aerosol, intranasal, etc. And as we're not naturally derived, we're synthetic. We start with 100% raw materials go in and 100% end product comes out. It's economical. It doesn't have expensive and environmentally hazardous waste. And we're getting ahead with this. So we talked earlier about a pipeline approach. When we think of sepsis, sepsis is caused by an underlying bacterial infection. It could have happened from a bump on the table where a bacteria got into your blood system through a breach in the skin. It could have happened through your lungs. It could have happened through urinary tract infections. Of course, we mentioned some 25% of patients started there. So we take the perspective of an antibacterial approach. So we've completed our phase 1 and phase 1b, 2a for a UTI or urosepsis as we demonstrate there. We utilize the compound as a topical agent for acute bacterial skin structure infections, so any topical type infection. And we're working with the US Department of Defense, having completed a preliminary phase 2 in Australia with now the US government for a phase 2 in the US. How do we work? What is this novel mechanism? So we basically enter the cell wall. Firstly, we're attracted to the bacterial cell through a proteinaceous attraction and binding to the cell. And we have a very specific effect on the energetics of the cell, particular to prokaryotic cells. And of course, there's no evidentiary effect on eukaryotic cells. And what that means in simple talk is we shut down the ATP synthesis or the beating heart and energetics of the shell of the bacterial cell. We shut it down in its actively growing form. We shut it down in its stagnant, non-growing form. And we also stop that Z-ring complex or cell division complex taking place. As we see, one of the components for cell replication is entirely disrupted and they cannot recover. This is it at a molecular level. So looking down the microscope here, we see on the left-hand side, E. coli, the little guys, and then the big guys, your eukaryotic cells. So your enemies, your E. coli, your eukaryotics, your ally. Now, in exposure at the same time with the presence of Recce 327, our primary compound, you'll see the bacteria are dead. The enemy has been disrupted. They're not growing. The eukaryotic cells are growing and multiplying, unencumbered, uninhibited, your allies are getting ahead. So that's really important for selective mechanism of attack. This is looking to the totally opposite gram stain from E. coli across to staph. For the microbiologists in the room will recognize staph is slower growing, so it's not as entertaining. But you've got the little cellular ones there, which is your staph bacteria, and your bigger ones, your eukaryotics. Of course, when you look on both screens, one in the presence of without Recce growing, the other with the presence of Recce not growing. Very, very quick way of seeing at a molecular level how things are occurring. This is looking at our suite of escape pathogens. So this is actually done by a leading laboratory out of San Diego. By example, most interestingly here, two things. One, practical concentrations, but two, activity counted in minutes, if not within an hour. in every single instance across every bacterial species at a practical concentration, you're getting some 6 log reduction. That's 99.99999% reduction in that bacteria occurring in that time. That's truly extraordinary. Importantly, of course, we work and keep on working with repeated use. If you wanted to compare us to amoxicillin, say, they stop working after 4, 6, or 8 repeat cycles. We work well as the first time, second time, third time, all the way through to a minimum of 25 repeat cycles. I say minimum of 25. I mentioned earlier I was a founding investor. This was an experiment that I funded, and as we started and lodged patents which were granted for this, we've since done it hundreds and hundreds of times over months and months and never seen any resistance, and we don't expect to see any resistance either. This is looking to how we compare to existing antibiotics in a broader form. So here's E. coli, of course. Here you've got your cefalexins, ciprofloxacins, colostin, it's pretty much poison, tetracycline, and of course our compound against E. coli. So what's obvious and interesting is all of the existing antibiotics have no effect in the first hour and a half there. That's not to suggest that they won't have an effect. They will have an effect, but they take a lot longer. They require a lot more concentration, which leads to comorbidities, toxicities, and similar down the track. We work really quickly. We work really broadly. And the faster you can stop an infection an infection in a patient setting, the faster, of course, you can get on to treating the patient in its totality. So it's really important to stop that bacteria in its tracks, and that's being demonstrated here. So this one shows, of course, against a non-actively growing bacterial cell. So these are the more challenging bacterial cells by way of traditional antibiotics. And in a fairly simplistic form there, we basically see that we have a rapid onset of activity when the others don't. Now we're talking IV. So we've dosed about 100-ish people. I think 80 active and the rest were placebo. But broadly speaking, we've demonstrated our compound over a 1-hour infusion to be very safe and very well tolerated. So you're putting some 4,000 to 6,000 milligrams IV infused into healthy human vein and looking for any signs of change in EKGs, telemetry, laboratories. They're all exactly the same. Here, you, of course, see a nice dose-dependent concentration in the human plasma as the compound is being recognized in the plasma. But interestingly, you see a significant, some 20-fold increase of the compound appearing in the human urine. Now, why would a compound be appearing in the human urine from a septic patient perspective? Well, it would appear that our compound goes from the blood through the kidneys into the urine, where, of course, we've demonstrated in follow-on studies that the compound is still active. So you've had an incredibly active, powerful agent that's been in the blood, it's passed through the kidneys, and it's appeared in the urine. You have the potential to treat the entirety of the urinary tract infection patient or the uroseptic patient from their underlying UTI all the way through to the kidney infection or sepsis itself. So does it work in urine? This is just another experiment by example. Some 6-log reduction there in 15 minutes. Very impressive. We've, of course, shown not only can we, because we're doing it so quickly, can we administer quickly? And that's to move away from just the hospital setting, which is an hour and over, to the first patient presentation setting, perhaps at the GP level. So we've shown in as little as 15, 20 minutes, in fact, it says 20 minutes there is the most optimal, that we can rapidly infuse this to get on top of that infection and stop it in its tracks. So now we're moving to the broader spectrum, of course, from the perspective of other causes of sepsis. So here we touch upon our topical program. We've completed a preliminary phase 2 study against topical burn wound infections. We've completed a preliminary phase 2 study against diabetic foot ulcer infections. In both instances, all patients had a complete clinical response. We've now gone, hey, it works topically. Let's expand the study, of course, into ABSSSI. And we have a current study in that area, which is progressing very well. Of course, we'll update in due course, but that's happening across Australia's largest leading teaching hospitals. The one you may not have expected is we're stepping into Indonesia. So why Indonesia? 11% of Indonesians have diabetes. Of course, if you have diabetes, you're twice as likely to get an infection. If you have dialysis, you're something like 100 times more likely to become septic. So our partnership, as we've demonstrated here with a leading Indonesian pharmaceutical company, and there we are with the Indonesian Minister of Health, has positioned us to enter the ASEAN region to treat the diabetic patients, of course, which may result in early sepsis or advanced sepsis studies taking place in that region. So the compound is really very much stepping out into broader regions, both the US under Department of Defense in due course, likely a phase 2-3 sepsis study there in the US and beyond. So how are we doing it? We make it inexpensively. So we have our own manufacturing capacity. It's a few dollars a kilogram of raw materials. Of course, that's 100% converted to end product. We have no expensive waste. We're producing in quantities suitable for all phases 1, 2, and 3 of clinical studies. And as you can see, we're scouting US locations at this moment to enter clinical trials in the United States, having successfully delivered them in Australia phase 1 and 2, which of course is accepted by the FDA as a FDA accredited study. We've got patents to say that our claims are a fact, so methods of manufacture, curative claims, preventative claims, and even some antiviral capabilities. If a virus has a proteinaceous cell, we target the proteinaceous layer of that cell and it appears then interact with the virus. So we received some nice US Department of Defense money. It's pretty foreign for a US government to be deploying tax money across to a foreign entity, but a foreign entity here in Australia received US $2 million to advance our burn wound program with the US Department of Defense. So we thank them for that and look forward to updating as data becomes available. In summary, this is a first-in-class, a brand new class, broad-spectrum anti-infective. We have a broad portfolio approach, targeted and focused upon sepsis, bacteremia, to be perfectly clear, which is the broad-spectrum nature of sepsis, including the underlying urinary tract infection. We treat the AAB-SSSI or topical infections that the patients with the all-out septic infection may have that underlying bacterial burden occurring from such a place. We have multiple phase 1, phase 2, and in fact, a registrational phase 3 study coming online. We're able to make our product, we're able to supply our product, and as World Health recognises clinically the most advanced new class of antibiotic in the world at this time, we really look forward to delivering the great promise of what we've been able to see here and a sign of new hope in the fight against superbugs in the spectrum of sepsis and the patient cohorts alike. I'm James Graham. This is Recce Pharmaceuticals. Thank you for listening.